Hello class, today we are going to start up with our topic, that is electrophoresis. Electrophoresis is an advanced separation technique that is widely used in laboratories as well as for commercial practices for the separation of particles. Which kind of particles will be separated? The particles are basically separated on the basis of their size, their shape, their molecular mass, and very importantly on the basis of their charge which are present into the system, the charge that are present over to the particles which are to be separated from the rest of the materials. How electrophoresis is going to separate? Electrophoresis is a technique that basically works on the principle of the separation of the charged particles in the influence of the electric field. How is it going to separate the system? The solutes that are to be separated are mixed into the system. These are basically mixed into the liquid which can be having electrolytes which are going to be moving either towards the positive electrodes or towards the negative electrodes. How this particular technique can be beneficial? If our system is having charged particles, the charged particles could be either positively charged or the negatively charged system. In the presence of the electric field, due to the electrostatic forces which are arising due to the charges, these will help such particles which are charged to move into this particular system. The negatively charged particle, as we know, will try to move towards the positive electrode, whereas the positively charged particle will try to move towards the negative electrode. This is the basic principle of this particular technique. As we consider the case, in a particular apparatus that is having two kinds of charges, that is positive and negative, if our particle is present, that is, it could be positive charge or negative charge. If this particle is negative charge, it will try to move towards the positive end. If this particular particle is positively charged, it will try to move towards the negative terminal. This is the basic principle that is employed in this particular technique, that is electrophoresis. As we know, electrophoretic apparatus that are present in our laboratory, it could be of two types. This particular diagram is showing the apparatus that is of vertical kind. It is basically used for purification and separation of proteins, whereas this one is the horizontal apparatus. It is now very commonly used for separation of nucleic acids. Now, how are we going to create the system that could be charged in any kind of the case? We are directly going to use either the intrinsic charge that is present into the system, or we are directly going to apply certain chemicals to certain chemical treatment to the system through which it could be getting charged. Like in the cases of proteins, as we know, proteins are made up of amino acids, and amino acids could be either positively charged, they could be either negatively charged, or they are neutral. So how are we going to purify such kind of proteins with the help of this system? In such system, we are directly going to apply a sample into this electrophoretic chamber. In this chamber, we are going to add electrolytes, and then we are going to connect them with the power supply. The power supply will be provided at the two terminus, the negative terminus as well as the positive terminus. Due to this, if the molecule is uncharged, it will try to remain at that particular site where the neutral charge is present into the system. That is, it will be stationary, it will be non-motile at any point. Whereas the charged particles will move on the basis of their sizes to either of the terminus, as we have already discussed. Next is, what are the techniques that this all part that we have discussed, it is limited only to laboratories, because it would be very much difficult for us to continue with the gel filtration for commercial practices. Why? Because gel filtration technique, or the electrophoresis technique will be very much time consuming. It requires a lot of labor, and the amount of purified that we are going to get will be very, very less. So there are two kinds of electrophoretic techniques, that are gel electrophoresis, and the other one is the free flow electrophoresis, which is also called as the liquid electrophoresis. Clear? So what is to be done in the free flow electrophoresis? We are not going to cast the gel, whereas we are directly going to apply the electric field into the liquid, and during the motion of the fluid, the particles will be separated on the basis of their charge. So first we are going to consider the case of gel electrophoresis. Gel electrophoresis requires first of all, we have to prepare the gels. Gel could be either made up of agarose, it could be made up of polyacrylamide, it could be made up of sucrose, or other kind of gelling materials. Gelling basically requires the principle of cross-linking of the polymeric compounds, due to which the concentration of the gelling materials will either increase the pore size or decrease the pore size within this kind of cross linkage is seen in the gelling materials. The more concentrated will be the gelling material, the lesser will be the pore size, and the lesser concentration of the gelling material will increase the pore size. These pores are basically important for the movement of the particles that are to be separated under the influence of the charges. What kind of charges? The charges that are bearing to them and the charges that are at the terminal due to the application of the electric field. In this particular system, how is it going to be used? We are going to cast the gel, we are directly going to make wells. Wells are basically made by the help of combing. Combs are simply made up of materials of which are gel casting tray and other materials of electrophoretic units are prepared. Gels are to be kept first, over to which the molten gels are poured. After pouring, we will have to let them stand by for some time so that the gel can solidify. After some time, we are going to remove the comb. After removing the comb, we will find that there will be some kind of gaps within this casted gel. These gaps are basically the site where we are going to pour our material that is to be electrophoresed. After this, we are directly going to switch on the power supply. This power supply will activate the two terminals, that is, we will be having a negative terminal and a positive terminal. In either case, either it is the protein case or in the negative or the nucleic acid case, in both the cases we try to use, for best results, the casting of the gel is done towards the negative terminal, so that majority of the particles are moving towards the positive terminal. But as we know that in the case of proteins, there are certain contradictions, like amino acids could be either positively charged or they could be negatively charged. In either case, we are not only having simply a single type of charge. So what are we going to do in such cases? We can treat them with chemicals like sodium dodecyl sulfate, that is known as STSPage. It is a short form used for sodium dodecyl sulfate polyacrylamide gel electrophoresis. In this kind of system, as we know that polyacrylamide gel is specific for proteins, whereas agarose is important for purification of electrophoresis of nucleic acids. For polyacrylamide gel electrophoresis, if we treat them with sodium dodecyl sulfate, how is it treated? We directly add sodium dodecyl sulfate in required amount and we are going to boil the protein sample. Due to this boiling, the protein will first of all uncoil. It will coil around the sodium dodecyl molecules and it will be ready with this. How is it going to be ready? It will be having a uniform net negative charge over a particular unit of that particular polypeptide or the protein. Thus, if the whole of the protein is negatively charged, then it will directly migrate towards the positively charged. But at times, when we are striving for the commercial product and that particular commercial product is not required to be treated with chemicals like sodium dodecyl sulfate, at those instances we are directly going to use the native page. What is native page? Native page simply means that we are going to directly use its intrinsic charge. Intrinsic charge means that we are not going to treat it with any kind of chemical, we are not going to alter its shape, its size, nothing. We directly are going to use the protein as it is present into the system, that we are not going to change its shape, neither we are going to modify its charge. At such instances, we are going to cast our wells exactly into the middle of this particular electrophoresis chamber, through which it could either migrate towards the negative terminal or it could move towards the positive terminal, as is the case of the protein. If it is positively charged, it will migrate towards the negative terminal. If it is negatively charged, it will migrate towards the positive terminal. So it is all about the electrophoresis. Which kind of electrophoresis? Gel electrophoresis. The next is the free flow electrophoresis, which is also called as liquid phase electrophoresis. How is it going to be done? In this particular case, we have chamber having the terminals present like this. In between these two charged terminals, one could be the positively charged and the other could be the negatively charged terminal. When the power supply is activated, before activating the power supply, we will be adding our light that is to be separated on the basis of their charge. The fluid will be continuously running through the system and the particles will be migrating on the basis of their charge. As we can see that this particular light has been discriminated into three kinds of system. If our product is having negatively charged, it will be deviated towards the positive terminal. If our product is neutral, then it would be present over here, and if our product is containing positively charged material, then it will be deviated towards the negative terminal of this particular unit, and over here it could be collected into the 3 separate chambers. It is for collection of negatively charged products that are separated, it is for neutral products, and it is for positively charged products. So this is the basic principle of our free flow electrophoresis, that is also called as liquid phase electrophoresis. Third thing, it is the most important technique that is suitable for the commercial practices, that is for industrial level. Why? Because unlike the gel electrophoresis, it is not having many complications. We are directly going to run our gel. It is directly applying the 2 kinds of terminals, due to which in real time the separation can be carried out, and we are directly watching our product to be separated, and there are very less problems faced in comparison to the gel electrophoresis. Thus it is the technique that is best suitable for commercial practices or for industrial practices. There are certain variants which are present with this electrophoresis, like there are isoelectric focusing or the 2-dimensional gel electrophoresis. This we will be considering. The first variant of electrophoresis, a common one, is the isoelectric focusing. It is abbreviated as IEF. In IEF, we try to create a gradient, that is the pH gradient. pH gradient means we have created a system into which the pH is either in the increasing order or it is in the decreasing order. There are various chemicals available with us through which we can create the pH gradient. Once the pH gradient is set up into a system, then we are directly going to pour our material of our interest over to this system. If once the material of interest is set up over to this system, it will try to sink down. During its settling into the system, the molecules present in our sample, they will be settling at a position which is having their pH equivalent to the pH of this particular pH gradient. Like suppose we have our pH 1, 2, 3, 4 like this. If the molecule is having its Pi value at pH 4, so it will be settling over here. If its Pi value is equivalent to 3, then it is going to settle over here. Now the question arises, what is this Pi value? Pi value stands for isoelectric point. It is that particular point on the pH scale at which the whole of the molecule will be having neutral charge. How is it going to be neutral charge? That either all of the components will be in charge in such a manner that they will be cancelling to each other, or the whole of the system will be acting in such a way that it is going to neutralize all the charges present on that particular molecule. Such kind of pH is known as the Pi, is isoelectric point for that particular molecule. The mobility in this particular phase for that molecule will be neutral due to the absence of charge. In actual, the charge is not completely absent, but it is neutralized due to equivalence of both the charged molecules. This is the basic principle of the isoelectric focusing technique. This technique has been in continuation with another technique, that is polyacrylamide gel electrophoresis, but these both techniques run perpendicular to each other. That is giving one more advantageous technique, that is combinedly known as 2-dimensional gel electrophoresis. An abbreviation known as 2DGE used for this kind of system. Let's see what 2DGE is. In this isoelectric focusing, we already know that the molecules will be separated on the basis of their pH gradient. This particular tube will be used with the another system on which our gel is casted. If this particular tube is containing the molecules which have been setted according to their density gradient, now this tube in connection to the gel will be connected in such a way that it will be connected on one kind of terminal only, so that the movement of charge, if during the isoelectric focusing was vertical, now in the second phase the movement will be in the horizontal plane. In either case, the isoelectric focusing as well as the polyacrylamide gel electrophoresis have to be done perpendicular to each other. Why so? Because it will provide a separation manner in 2 separate dimensions. One will be in the horizontal manner and the other will be in the vertical manner. Like it is in the vertical manner, and the gel electrophoresis will be done in the horizontal manner. Now if these particular molecules have been separated based on their pH gradient, now they will be separated based on the charges and the mobility of the charged particle into this electric field. Like suppose this particular band that is having molecules that is separated on pH 1, now they will be separated based on their mobility into the charge. Some molecules will be completely neutral, they will be standing over here, rest will be starting moving based on their charge intensity. Like this particular band will move like this. So it is basically giving us the separation in 2 different dimensions. So this is the basic advantage of this particular technique. Again the problem is this, that this particular technique is very best for analysis purpose, but it is not very much helpful for the industrial purpose, industrial purification practices. So this is all about the electrophoresis. Next comes that how a nucleic acid is going to be separated. For nucleic acid we are directly going to use the gel electrophoresis system, in which a horizontal gel is casted. We are directly going to use this particular vessel in which the gel is first melted and it is settled down. We are going to put a comb which is going to prepare the gel, which is going to prepare the wells. We are going to remove the comb after the solidification of the gel. We are directly going to add our product that is to be separated, that is a solution containing nucleic acids that are to be separated based on their size, charge, shape, etc. As we know that nucleic acids are DNA and RNA, these both contain negative charges, so they will be loaded onto the negative terminal, and after the application of the power supply they will be moving towards the positive terminus. Next important thing is that we will have to add buffering agents. These buffering agents will be containing the electrolytes. Due to this, their mobility, this particular thing will be separated. Same is the principle for the protein separation. For proteins we are not going to use the horizontal gels, we are going to use the vertical gels, because vertical gel will provide one more efficiency, that is the sinking of the protein due to the motion of the electrophoresis. As we have seen that for protein purification the capillary size is very small. The gel that are casted, these are of capillary size and those are very small. Thus the vertical system is much more beneficial. It is beneficial for loading practices. It is also beneficial for the movement of the protein into that particular system. So this is all about the electrophoresis technique, which is a very important separation technique, not only for the laboratory purposes but also for the commercial or the industrial purposes. That is all. Thank you.